I think we should go ahead and start. So today's my pleasure to introduce Dr. David Wong. He's the Felix and Mildred Eep Endowed and Distinguished Professor. He got his DMD in dental medicine from University of British Columbia, Vancouver. And he was a professor and head of the Division of Oral Pathology at Harvard School of Medicine, Dental Medicine. And since 2002, he's been recruited to UCLA School of Dentistry. And he is the Associate Dean of Research for School of Dentistry and the Director for Center for Oral Head and Neck Oncology Research here. And David, his research has focused on basic and translational sciences for oral cancer, really revolutionizing the use of saliva samples for development of salivalomics, exosomics, and diagnostics. Really cool. He has applied his biomarker approaches to basically identify biomarkers for oral and systemic disease, such as oral cancer, pancreatic, gastric, and lung cancers. His lab has developed this efirm liquid biopsy test used to detect circulating tumor DNA in lung cancer. And so I can think of no one else that is more fitting than he is to develop approaches to identify COVID antibodies from saliva samples. So please take it away, David. We're really looking forward to your talk.
Will do, Melody. Thank you. Thank you. I'll go to screen share here and pull up my slides. Thank you for having me here, and thank you all for coming in on this Wednesday noon hour in this very special time. Unusual, unsettling. Okay, all right, Melody, do you see the slide coming up?
Yep, it looks great.
Fantastic. So again, thank you for having me here to speak with you and share with you what we've been doing since March, and it's a very special calling, and really appreciate that opportunity. I've been enjoying these seminars, Melody, and all the colleagues on the line last week. Jason, your talk was just brilliant. Just to know of the breadth and depth of the expertise and the enthusiasm. It may not be a good word, but you know what I'm saying here in terms of this campus is coming together and repositioning, repurposing, if you will, doing things that we weren't doing 6 months ago. I want to take the time today to share with you a portfolio of research that really is echoing the call for testing for this pandemic, and the utilization of the clinical sample type that could provide the capability for population level testing, household to household, and at the point of care, and the eventuality of identifying whether an individual is infected, and in addition to development of host immunity. So the project was 2 weeks ago. It is currently being funded by the School of Medicine, the Stem Cell Institute, and we're also glad that 2 weeks ago was selected as a Red X project. This is my disclosure. And the current support to the lab. So I want to utilize the time today, as you know that the topic really is recent, but is timely in a sense that what we do really align in terms of this mandate of this national global importance of testing. But I also want to sort of echo a thought that Melody talked about is sort of the use of saliva as a sample type for testing, and the historical aspect from a scientific, translational, and clinical, how sort of it's been advanced to this juncture for SARS-CoV-2 detection. So if it means anything, is that what we've been told is that since the Red X initiative was announced at the end of April, there were over 3,000 proposals submitted, and apparently 35 were selected. The point is that half of these funded proposals or submitted proposals was saliva based. So I think it's something that needs to be taken to heart, Is it because there's no alternative capability? I think the answer clearly is not. But perhaps it's sort of, and it's not an accident. Perhaps I think that's a good way as we look at it. And I like to share that perspective with you in terms of the science, how it developed from where it was, and how do we come to this juncture. So 14 years ago, the National Institute of Health, in particular the Dental and Craniofacial Institute, implanted into the scientific community two initiatives. One is to comprehensively decipher the salivary proteome, so that these proteomic constituents would be utilized, in terms of understanding biology, but importantly as a diagnostic alphabet. So biomarkers development and could be begin to sort of consider in utilizing this clinical capability. The other is to develop point of care capabilities that clinicians, physicians, nurses, and dentists of the future could utilize, a clinical sample type saliva, and then could evaluate these omics constituents for early detections of diseases, monitoring, treatment. So the marriage of the proteomic catalog, if you will, and these point of care tools really constitute a capability that the NIH sort of projects into the future. So that's sort of is where it set the stage of the work. When we got involved with this landscape, it is important to us. It's clear to us that for saliva to advance itself to a clinical reality, there's this true frontier that needs to advance and also converge. One is a translational and clinical frontier where biomarkers that needs to be developed, be it proteomic, genomic, metabolomic, that needs to be developed using guidelines that are robust. The NCI EDRN has set forth those, and then advance it towards clinical utility. The other is the scientific frontier, that in particular why a systemic disease, when it developed, there could be these omics constituents that are reflective of the organ of pathology that are traversed through our body and come into the salivary gland, into the oral cavity, that could be harnessed, trained, and advanced forward translationally. So a quick sort of overview in terms of where saliva comes from, that perhaps many of you are kind of wondering. So saliva is produced mainly by three major pairs of salivary glands, the parotid and the salival cheek, and two pairs that are sitting underneath our tongue, the sublingual and the submandibular. And on a daily basis, I just want to raise this question here. Does anybody happen to know how much saliva we produce on a daily basis? You can raise your hand. And here is a reference. Here is a bottle of water, 500 milliliters. How many volumes of this do you think, a fraction of it, do you think that we produce on a daily basis? Anyone? Melody, you see who raise a hand, or just shout, and anyone care to venture an answer? Okay, hearing none. 600 milliliters. So this is 500. So basically this plus 100 milliliter a day, each of us produce non-invasively, non-painfully, non-embarrassingly. It comes in the oral cavity. And I'll submit to you that it also carries all these omics constituents that are being utilized for personalized medicine, precision medicine utility. So it has that endowed capability, and it's coming into, and it's a volume that could be obtained repetitively, continuously, and non-invasively. So the next slide is important because it demonstrates that the salivary gland apparatus is a typical one, the acinine cells and ductal cells, where the saliva comes from, and perhaps there is a communication or channeling of constituents from the vasculature that comes into the salivary gland. So this slide here demonstrates that the vasculature that nourishes the salivary gland, this is a typical salivary gland, acinine cells and ductal cells, they're active, passive, and secretory mechanism that pulls these constituents from the vasculature into luminal space of the salivary gland and into oral cavity as saliva. this schema really explains why, 16 years ago, Charlie Strickfus at University of Mississippi that found that HER2 neu, a breast cancer marker, can be identified and utilized as an early detection capability. It really caused quite a bit of attention. And so this schema here begin to scientifically, underpin that capability. One thing of note that we should pay attention to is that while we see the content of circulation do come into the salivary gland, but the content that's produced by the acinar cells or in the salivary gland apparatus do not go retrograde. They do not go from the salivary gland into systemic circulation. So this would become important as we begin to look at mucosal immunity. Should the salivary gland, which is known to have ACE receptor and pathogens, encounters and local plasmacytes that begin to produce immunoglobulin, especially IgA, secretory IgA, if they were produced, they would be entering the oral cavity rather than going into in a retrograde into the circulation. That has scientific and clinical importance, because if saliva could be used to monitor, the emergence of host immunity. Okay, so and if secretory IgA is an important reporter, if you will, not only biologically, you can begin to think about neutralizing the virus and amongst others. The oral cavity could be could be a venue for evaluating that. So we begin to paint this picture with leadership on campus that Saliva Omics Knowledge Base. This is a resource that initially was housed in chemistry and now is housed in school dentistry, where genomic, proteomic, and other constituents, inclusive of point-of-care tools that enable this capability, are being developed and open to the public for utilization. This are the leadership that we work with. Joe Liu, Kim Fo, and Julie Weilich, and many of you know and work with, together with John Yates and Susan Fisher at UCLA, we comprehensively catalog and decipher the entire salivary proteome, and this was about 12 years ago, and there's 1,100-ish protein, and if we stratify them into the parotid fluid and the sublingual fluid, we can see that about 2/3 of them are common, and yet 1/3 of them is uniquely present in the parotid fluid and in the sublingual fluid. So this capability is in place, and the catalog is well deciphered. The list continue to grow as we can imagine as technology advances itself. The other omics constituents, the transcriptome, the extracellular RNA, take on a slightly different path of development within saliva. Now, viral RNA, that's where the Rutgers test is. That's where the Yale test is, as we heard this week. And the rationale for that, if anyone were to be asked to all of you, a few years ago, you probably would frown and say, how could RNA, survive in this environment where it's exogenously communicative, and also nucleases are actually known to be present in saliva. So when we published that this almost I'm looking at the year here, 14-ish years ago, 2004, actually 16 years ago, it was met with quite a bit of concerns, rightfully so, but that's sort of led us from where we are to Harold Martinson, which many of you know, who was the chair of chemistry at the time, and clearly, leadership in RNA, biology, molecular biology, and we brought forth a number of scientists to our lab to scientifically credential the existence of RNA in saliva. So that escalated into a number of basic and translational utility. But perhaps what is most gratifying is that 7 years ago, when the NIH Common Fund began to unfold a program called Extracellular RNA Consortium, whereby RNA is no longer considered, it has to be confined within the nucleus of a cell, and move to cytoplasm for translational. They could be secreted by a host cell, encapsulated in vesicular editing, exosome in particular, that could traverse from an anatomical location to a distal target, uptake by the target cell, and translate into a proteomic constituent that could influence the physiology of that target cell. It's truly a new paradigm. The ERCC now is in its second phase of advancement. Again, still a common fund project. And again, so looking back 14 years ago, having that initial discovery here on this campus and then leading to this sort of paradigm changing information or knowledge base is gratifying. So the question is asked, is that RNA of these coronavirus being detectable in an environment like saliva? Is it surprising perhaps, or is it there's a historical perspective to it? And I'll come back to that point shortly. So working with Ray Schall, who's a scientific leadership on this campus here, we begin to really take a deep dive. Quite a few years ago, comprehensively deciphering the RNA constituents in saliva. Again, this is part of that ERCC initiative. Amongst the many intriguing outcomes is the presence of this non-coding RNA, the circular, the snow, and amongst other, we're looking at here a profile of the peri-RNA in saliva and how it cluster with other body fluid and cell type, and they caught our attention because saliva cluster very closely with the embryonic stem cell, and skin suggestive of a proliferative capability. We're currently putting that to test in a very unique environment of wound healing in the oral cavity, which is known to be much more rapid than skin wound healing. So these capability also allow us to begin to take a new look at this horizon of biology within the oral environment, because previously, very little is known, and as we all know, extracellular RNA, non-coding RNA, and the center stage that they are taking a role in understanding biology is really paramount. So the extracellular RNA consortium now have in its public domain the extracellular RNA atlas, and what we can see here is saliva clearly has embedded its footprint within the biological fluid in terms of extracellular RNA. These here constitute the small RNA, and there is still an equal number of long RNA that needs to be input, but we're very glad that it makes its presence and it makes its footprint within the extracellular RNA consortium. So with that in mind, the third constituent perhaps of these omics targets within saliva is genomic DNA circulating tumor DNA, and in that regard, I'm glad to share with you that we have brought forth saliva as a clinical sample type to the NCI Liquid Biopsy Consortium, together with colleagues at Hopkins, MGH, Yale, that circulating tumor DNA, which are pathognomonic of human cancer. Case in point, non-small cell lung carcinoma. If somebody have a sensitizing mutation in EGFR and A50 and exon 19, okay, so unfortunately that individual would have non-small cell lung carcinoma. So we were very, very, very fortunate to be working with Dr. Denise Aberle in radiation biology, Steve Dobenet in pulmonary medicine, and then David Alshaw, which I think many of you work with, and also an industrial partner, Liquid Diagnostic, Dr. Charles Strom, who is a veteran from Quest Diagnostic to advance his portfolio. This is the technology that I would want to share with you today that really takes center stage in terms of this REDX initiative. This is an electrochemical technology whereby a drop of biofluid, be it plasma, be it saliva, and in the presence of electric field, did I lose my sound? Can you still hear me?
We can still hear you. There was just a little kink, but you're fine right now.
Okay, all right. So if it's not coming through well, just let me know and I'll do a few adjustments here. So a drop of biofluid in the presence of electric field, these omics constituents within vesicular structure are released, and they are detected in the presence of a hybridization schema in a high throughput format. where in this case here is a circulating tumor DNA with a mutation, and this mutant target is detected in a dual manner. These capture probe, which identify or capture the mutation annealed, and then a detector probe with a reporter molecule.
You've reached voicemail of Dr. Elise Singer.
It also needs to sit on this target. So there's a dual layer of specificity. Both need to sit on it and then come in with the substrate TMB, binds to horseradish peroxidase, and then elicit electrons registered by the GO service. And this is recyclable in the presence of hydrogen peroxide. So this is a very streamlined. The entire process is about 15 minutes, and it requires a microliter worth of the biofluid. And that becomes important because the most recent EUA test for RNA in saliva took on that importance, bypassing sample processing, because if they don't, then pulling out RNA from saliva is just inconsistent. You lose the sensitivity. So that Yale test that came out literally days ago bypassed that. I think the NBA endorsed it amongst other sports venues, so to speak. So the performance of this platform is demonstrated here in 2 blinded clinical studies coordinated by EDRN at NCI, and we're looking at non-small cell lung carcinoma individuals in their plasma before and after biopsy. What we see here, those individuals with the exon 19 mutation or with the 858 mutation, these 2 mutations are mutually exclusive. You have one, you don't have the other. What we see here in this cohort of 44 subjects, those individuals with exon 19 are clearly detected. The other mutations is at a control level, whereas the other detection of the 858 gives you the opposite observation, so to speak. That's plasma. This is blood. We can do digital, we can do NGS on this. The concordance is about 70%. What we see here with efirm is embarrassingly, almost perfect. Okay. Now that's plasma. In saliva, this is what we see. Again, this is a blinded clinical study coordinated by NCI. And what we see here is equally discriminating ability for efirm to identify exon 19 in red and 858 mutation in saliva of these same patients. These are pair samples. You have saliva and plasma from the same patients that have clinically diagnosed non-small cell lung carcinoma genotype at the hospital, blinded, sent to MD Anderson, then sent to us here at UCLA. Then the test was run. According to our statistician, the discrimination in saliva is actually cleaner. You can see that distinction between the mutant and the control level. So it was a technology that came into place. I think at the time was 2014, a mere 6 years ago. And this is sort of what we're excited about in terms of liquid biopsy capability. Advancing it forward, where the laboratory technology needs to hit that next milestone of translating into clinical regulatory ability. So we're very, very fortunate indeed on this campus here, their leadership and perhaps receptiveness that would work with us and entertain saliva as a sample type. So Dr. Wayne Grody, Josh Nagan, Omar, Gardner, and Lily Zhang has been working, with us collaboratively, in molecular pathology, in clinical microbiology to advance this platform. And this is a reader where we can have a plate. This is an electric plate, a 96 well plate, where on the bottom are these electrodes that does this electron registration, if you will. And this is placed into this reader here, and within minutes it will have a readout. This is the laptop that drives the entire system, and then a plate washer. That's the entire footprint. That's the entire footprint of the efirm capability is now placed in molecular pathology. And what I'm glad to say is that working with Dr. Grody and Dr. Josh Dagan in the last 2 years, we have obtained clear qualification a year ago in March for plasma, clear evaluation, and 2 weeks ago for saliva quantitative evaluation of these baseline sensitizing mutation in non-small cell lung carcinoma. So the process has reached an important milestone that this is no longer a laboratory technology, which is good, but it now has reached a juncture of being deployable, in a clinical setting in a reference lab, and I'll come back to this shortly as well. Then came March of this year. This is the dashboard from Johns Hopkins, which we're all seeing, and so it's clear that the development of therapeutics, vaccine and the ability for testing is still a soaring need, globally, and the ability to have those tests, deployable at the population level, at the point of care at home, and is paramount. So we know these are the EUA tests out there for detecting viral infection, either for the viral RNA or the viral antigen, nasopharyngeal swab, and more recently from Rutgers, the saliva test, and the antigen test from in saliva as well, from Quidel, and 2 days ago, the Yale test, which is a saliva test as well, but they sort of bypass that sample processing, which is an important aspects of it. This is a demonstration. I think this is the saliva direct test here. This is saliva direct test that came out a couple of days ago. So we know about this for since March, and obviously since that time we're glad to see that saliva is being utilized for detecting the viral genome. We're also glad to see that saliva being utilized for viral antigen detection, and perhaps as researchers, we're just glad that this is happening, and there are industry that wraps around it and wants to bring it for clinical deployment, and that's terrific. But that's one thing that hasn't happened yet in saliva, and that is serology. Okay, we know of these tests that's been rolled out. Many of them were questionable, and so the question is that could host immunity be detectable in saliva? As of now, as we know, it has not been deployed. So here comes where we came into our entry point of what we do, and I want to share that with you because it's important. So 11 years ago, after we did the salivary proteome, Dr. Gil Lohman at the University of Michigan, who oversees HUPO for the plasma proteome, reached out to us and say, David, why don't we do a comparison of the plasma and the saliva proteome? Makes great sense. Reach out to my colleague Joe Liu, Kim Fo, and Wei Hong Yang, who's the first author on this paper here, where after we did that, of the many things that were intriguing, one thing stood out in our mind. If we look at the immunoglobulins in the plasma and in the saliva, be it whole saliva, parotid, or combined submandibular saliva, the linearity of the IgA, IgG, and IgM is amazingly linear. Now, it's not target specific. It's not target against actin or any target, but as a whole, immunoglobulin isotype, they're very linear. Okay, so that was in our mind, and that was actually what's put to test as a corollary 2 years ago, that in a chronic autoimmune disease, Sjogren, which many of you know about, that one of the classical diagnostic criteria is detection of these autoantibody in the serum of these patients, which 9 to 1 are women, and there's 4 million Sjogren patients in this country. So we put that to test, and we'll show you the next slide, which is very illustrative, that led us to what we do now for COVID-19. If we look at these patients that have bona fide classified Sjogren, which we know that they have these autoantibody in their blood, which are these patients here in red, okay? We simply ask the question, can we see these patients and of these autoantigen in the saliva? And what we see here is literally every subject lit up in their saliva. And this is efirm as well. What is important here, efirm really stratify these data points in blood and also in saliva. Now, that's the point that I just want to share with you quickly, because at this juncture here, we're asking ourselves, serology has not been evaluated in saliva. And knowing this, I think we should look into it, because if testing at the population level, if the ability to concurrently evaluate immunity, viral infection, and viral load in the same sample type using the same technology, that would have value. That really would have value. So that's what we set out to do. We obtained, essential research status. The lab turned into BSL2+, and working with clinical, infectious disease leadership together with industrial groups, molecular pathology as well. So here are the early data that demonstrate the reflections of host immunity in saliva that are being evaluated in blood, but has unique feature and perhaps kinetics in saliva that we look forward to deploy and integrate into this RADx capability. So we build this assay. This is the go surface of efirm, and we anchor onto it either spike or receptor binding domain, and then test it using either the monoclonal antibody or patient sample, and then a report of secondary comes in. So this is the very first experiment that we did, literally very, very first. So Charles Strong is a veteran from Quest Diagnostic, a pediatric geneticist, 16 years at Quest building assays. This is really an expert in assay development, and with his collaboration and using this efirm assay, what we see here is that in 3 recovered COVID-19 patients, serologically positive, they have detectable IgG to S1, to anti-S1 in their saliva, these 3. And these are control subjects here. This is the very first experiment literally at the beginning of April. These recovered patients and their saliva, specificity demonstrated on the slide here. If we spike in authentic S1 into the saliva of these patients, is titratable. It's attenuation of the signal here, as you can see in patient 1, in patient 2, really demonstrating the specificity of the detection process. And that's gratifying, and to know that capability, and also the assay has this performance. Again, this was a question really is kind of in the known, but based on the work with Dr. Gil Lohman and also with the Shogun patients, really led us into this exploratory path, and really lit this energy and catalyst to move forward. Then we have the tremendous great fortune to work with UCLA campus infectious disease leadership, Dr. Otto Yang and Jennifer Felcher in infectious disease, looking at acute symptomatic individual and also recovered patients to evaluate their saliva in a more laid out, comprehensive manner. And what this slide here demonstrates to me, one of the most important findings. Seromno IgG, IgM clearly has a profile 2 weeks after onset of symptoms, and the kinetics we know up to 2 or 3 months. The profile of IgA is still emerging, and as I mentioned earlier, secretory IgA as part of the mucosal immunity could be of interest, especially when you consider this actually could be a portal of entry for the pathogen as well as the eye, the nose, and the oral cavity. What we're seeing here is these are patients, from Dr. Felcher, that in this patient here, year 1, what we can see is the detection of IgM, IgG, and IgA in this individual here, 7 days after symptoms. These are 2 recovered patients from Dr. Yang at this early juncture, and the IgA in saliva is also inhibitable, titratable, attenuated by spiking in S1 into the saliva. This was important. So all 3 isotype of the COVID-19 antibodies detectable. This slide here demonstrates the concept, what I call so far is immunoequivalency of distribution of these immunoglobulin in COVID-19 patients in their circulatory compartment and in their saliva. These are recovered patients from Dr. Otto Yang. What we're seeing here is that these 6 patients, with the exception of patient 19, where we clearly see a disconnect between the seromno level and the saliva level. The other 5 individuals, their seromno and the saliva level of anti-receptor domain IgG assay using an ELISA in Dr. Yang's lab, and assay using efirm in our lab. It's amazingly equivalent, if that's a good word for it. So that was important to know. There's immunoequivalency, between the two compartments. This reflects perhaps, the underlying, physiology. We also look at, using different platform of looking at using ELISA and also efirm and detecting these isotype, in the plasma. There's good concordance. There's really good concordance suggesting that efirm and ELISA are both likely looking at the same target. And finally, at this point in time, is that the efirm capability in saliva and also in the plasma, looking at a antigenic domain, is highly concordant between the two compartments as well. So putting it together, we can begin to project an enthusiasm where saliva, in addition to circulation, could be utilized to evaluate the profile, the kinetics, and also importantly, ultimately the biology of the onset, either through a systemic entry point, or through a saliva entry point where mucosal immunity could be addressed. So I'll sort of put this together in terms of where we came from and why is the enthusiasm of integrating these immunological capability. This is a drop of saliva where we know the virions are there, the RNA is shed. There's currently clinical EUA tests to evaluate those. What I share with you today is that host immunity is being reflected in saliva as well, and so these three triad of information could be accessed through a single sample type, where it could be obtained non-invasively, repetitively. It could be evaluated. It's direct detection, and I think we knew it for almost a decade with efirm, but I think it really comes into relevance in terms of COVID-19 because the ability to evaluate viral RNA and without having to pull it out is of paramount importance. Industry dropped their EUA tests simply because it becomes inconsistent, and the ability to bypassing that performance. It really is the message at the end of the day here that we're enthusiastic about how it came together. In addition, the entire schema, how sample could be collected at the point of care, has been developed and could be integrated into this deployment capability, where saliva could be collected at the point of care at home. A collector has been developed by my lab 6 years ago, and Samantha Chen is a DDS PhD student at the time, and it could be collected, self-collected, inserted into a barrel, expressed into 2 collection tubes, one for RNA stabilization, one for proteomic stabilization. You can see the date here. This actually is 2014. Sent to molecular pathology here at UCLA at ambient temperature. No dry ice, no ice pack, and the plate-based schema you saw before, and the footprint of the entire efirm capabilities here as well. These are the advantages that we look forward to advance and really mature quantitative. All the serology tests out there, as far as we know, are qualitative, which is still good, but this is quantitative. Remember that chart of Sjogren and how it really stratified out these individuals, on that efirm scale. It's clear ready. It's EUA ready, and it's deployable in 6 months, and it's scalable. This minimal risk to healthcare providers. Is a saliva sample is critically important. So on that note, I think the idea is for us to advance these capability, the ability to evaluate viral infection, viral load, as well as host immunity in a single sample type, where sample collection, deployment, and a reference lab capability are here. They're here on this campus, and we are now at the doorstep of clinical microbiology with Dr. Omar Gardner for an EUA evaluation of the saliva IgG test. Literally, that likely can happen in the next 2 weeks. So thank you for listening. These are the people that just really came together, in a most selfless way, and it just it's very heartfelt that pathology and people in my lab and liquid diagnostics, and again, this work couldn't have done without the former dean and current dean of the School of Dentistry. Immensely thankful to the School of Medicine, the Stem Cell Institute for supporting us, the work now that has led to this RedDex initiative. So on that note, thank you all very much for being here. Looking forward to working with you, and thank you. Mm-hmm.
Great, thank you so much, David. It was really impressive work. I guess we'll open the floor for questions. You can either unmute yourself and show yourself to ask, or you can type it into the chat room. And I guess, David, I'll ask the first question.
Sure.
So it's really nice correlation between the saliva and the plasma samples. I'm just curious, for that one patient where you could not detect the antibodies in the saliva sample, did you go back and try to figure out, is there a window of time after infection that will be the best time frame for catching antibodies in saliva?
Yeah. Melody, that's a very, very important insight. The short answer, we don't know. Accessing the sample has been a challenge and will continue to be, but we're mapping this out with Dr. Felcher and Dr. Yang, how we could begin to delineate. That sample has been assayed at least twice, and so according to Samantha, who maybe she's on the line, she can answer the question. But we simply don't know proteomic degradation, amongst other things. But I think that window of optimal detection is important. Now there's a stabilizer that actually in this collection tube that we're enthusiastic about, that could concurrently, it's always been a vision of ours, the ability to not only procure the sample type, but concurrently stabilize these major omics constituents, genomic, proteomic, and RNA, and then allow for ambient temperature transportation, from California to Massachusetts and beyond. Because if you assemble those capability, Melanie, it really puts a clinical deployment in a different level. Because if you have to use dry ice pack, and pull out targets, and then collect each of these targets using a special collector, it's still good. It's still good. I'm not saying, but sort of, I guess if we could assemble all of that and integrate it into a schema, I think that really is the holy grail.
Mm-hmm. No, that's really impressive. I mean, it just makes sample preparation so much easier and convenient. Oh, and then Leah was saying that maybe you can stop sharing your screen.
Oh, sure. Okay. All right. Sorry. Oops. Let me exit here, and great.
Okay.
So,
any other questions?
Yeah, I have a question. Thank you for such a wonderful talk. I think it's really exciting that hopefully we'll have some really good diagnostic, high throughput tests coming through soon. There's been a lot of, concern about the fact that the serologies that we're seeing in the blood and the serum seem to be so variable from patient to patient. And so we really, have not been able to use serologies to make any kind of diagnosis right now. And, we're stuck with the nasopharyngeal PCR test. And I'm wondering if you think that the saliva will end up like maybe IgA, or whether it will end up being more consistent as a diagnostic test in terms of a serological test.
So I think if one thing we can show is that most of the serology tests are qualitative. Okay, so give you a yes and no answer, which is good. Okay, but it's clear to us that efirm has that quantitative, and that's the test we're heading for evaluation with clinical microbiology, and so I think that in itself is important. But as you know, the antibody is really reflecting on a host response versus a viral infection, and it just conveys these two levels of information, which could be important. I would think that if there's an ability to assess all three levels of information in the same sample type repetitively, I would do it. I would. Okay, and if it's easy and it's inexpensive, if I could collect the samples at home, I don't think anyone would have any issue with it. And it has to be good. I think that's that really is the ultimate benchmark, which I clearly learned from our pathology colleagues, and that's what excite us is not the word, but that's what is important that this can deliver. Now here's the thing too. So evaluating antibody in saliva is somehow, for a reason that we cannot put into exact context yet, ELISA Luminex is plaguing that. It's not coming through. I'm sure there are people that will come up with claims, but I urge you to look at the data. They're very compressed. They don't spread out, and at best is a qualitative test. That's what we see in Sjogren, and I can be certain that the sort of this will be repeating itself in this COVID scenario as well. Well, if there's no further question, I really want to thank you all, Melody and Bridget, and I want to thank everyone on the line, and wish everyone well and safe, and I really appreciate that opportunity and the breadth and depth of scientific leadership and expertise on this campus is immense. So thank you, Melody and Bridget, for having me here, and thank you all for coming online today. Thanks very much.
Great. Thank you so much, David. We're excited about the launch of this new test. And just a reminder to everyone, we will not have a seminar next week, but we will come back on September 2nd. Chris Seat will give a seminar. So see you in, I guess, 2 weeks.
Thank you.
Bye-bye.
Bye, Melody. Bye, everyone.
Thank
you. Bye.